Okay, so deep inside every single one of your muscles, there's a gigantic molecular spring. Now, for decades, scientists have been stumped by a really weird puzzle. What happens when that spring doesn't actually break, but just goes slack? Well, the answer is this incredible story of molecular engineering that's finally unlocking the secrets behind some really devastating muscle diseases.
So let me ask you this. What do you think happens when the biggest spring in your body breaks? You're probably picturing something sudden, right? A dramatic snap. But the reality is actually a lot quieter and way more insidious. And figuring it out has unlocked the secret to a whole category of muscle diseases. So let's dive in.
All right, first up, muscle's giant spring. To really get this puzzle, we first have to get to know this absolute biological titan. I want you to meet Titin. And when I say Titin, I mean it. This is the largest protein in our bodies. It is a true giant. The best way to think of it is like a molecular bungee cord that's woven right into the fabric of our muscle fibers. It's the reason your muscles have that passive tension, that springiness, always ready to recoil.
Now, titin isn't just kind of floating around in there. It's a critical piece of the architecture. It actually connects the key parts of the muscle's tiny engine, which is called the sarcomere. So with every single movement you make, every stretch, every step, countless titin springs are stretching and recoiling, literally holding the entire engine together.
And that right there brings us to the core problem. For years, we've known that if you have a mutation in the gene that makes titin, it can cause a whole range of really severe muscle diseases. We call them titinopathies. But the big question, the how, has been a complete black box. Why do these little genetic changes cause muscles to literally waste away?
So, to really appreciate the breakthrough we're about to talk about, you have to understand why this problem seemed impossible to solve for so long.
Here's the big scientific dilemma. Titin isn't just a spring. It does other jobs too. It's involved in cell signaling, it acts as a scaffold for other proteins. So if you just get rid of it completely, you lose all of its functions at once. It's like trying to figure out if your car's problem is the engine or the wheels by just blowing the whole car up. How could you ever prove that it was the loss of springiness alone that was the real culprit?
And this is where the absolute genius of the solution comes into play. You see, the old method, what's called a standard knockout, was a blunt instrument. You take out the whole protein, and you're left guessing which of its many jobs was the important one. But this new technique, the mechanical knockout, or MKO, it's like a scalpel. The protein is still there, doing all its other jobs, but it's been snipped in one precise spot. The spring goes slack, and only the mechanical tension is lost. It perfectly isolates the one thing they wanted to test.
So let's talk about the incredible tool that made this possible. This is the story of the molecular scissor, a true breakthrough in genetic engineering.
The first step was just brilliant. What the researchers did was create a special strain of mice where the titin gene was just slightly tweaked. They inserted a tiny, very specific sequence of code into the protein, basically a little sign that says, "cut here." They literally painted a target right onto the titin spring, a target that only one specific enzyme could recognize.
And the whole process is just so elegant. It unfolds in 3 steps. First, you have the engineered mice with the cut here target already built into their titin. Second, you use a harmless virus to deliver the scissors, a specific enzyme called a protease, right into the muscle you want to study. And finally, step 3, the muscle cells start making these little scissors. They find their one and only target on the titin molecule and snip. The spring is severed, the tension is gone.
So, the trap was set, the scissors were delivered, and the spring was snipped. And the consequences, well, they were nothing short of dramatic.
The very first result they saw was an absolutely catastrophic loss of power. I mean, within just 7 days, the muscles with the snipped titin could only generate about 1/3 of the force of the healthy muscles. This isn't just a little bit of weakness. This is a fundamental collapse of the muscle's ability to do its job.
And the physical decay was just as shocking. After only 3 weeks, just 21 days, the affected muscles lost a staggering 38% of their mass. They were literally wasting away. That's a condition called severe atrophy.
And this timeline just shows you how terrifyingly fast the whole collapse was. By day 3, you could already see the muscle's internal engine, the sarcomere, was starting to fall apart. By day 7, right alongside that collapse in strength, the whole organized structure was just gone. And by day 21, many of the muscle fibers were basically just empty shells, their engines totally disintegrated. All because one spring went slack.
Okay, so this is way more than just a fascinating lab experiment. This is the part where we connect the dots, from what they found in the lab all the way to the heartbreaking reality of human muscle disease.
So the researchers did something else that was really smart. They ran a milder version of the experiment. This time, they set it up so only about half of the titin springs could be cut. Now, this is much closer to what happens in a lot of human patients, who might have one good copy of the titin gene and one mutated copy. And the result? They saw a milder form of the disease, but one that looked remarkably similar to what you see in people.
And I mean, the similarities were just undeniable. When they looked under the microscope, the mice muscles were showing all the classic textbook signs of human muscle disease. The internal engines were a mess, key parts of the cells were in the wrong place, you had scarring start to form. It was a near-perfect copy of the human disease, and it was all triggered just by cutting the tension in titin.
So here's the crucial takeaway. For the first time ever, we have a clear trigger. It's not the absence of the titin protein that's causing the disease, it's the presence of a slack titin protein. That simple loss of mechanical tension is the first domino to fall, the event that kicks off this whole devastating cascade of muscle destruction.
And that brings us to our final, and maybe most important thought. This amazing breakthrough doesn't just tell us how the disease happens, it completely changes how we might think about treating it one day. So if a simple loss of mechanical tension is the trigger, it raises a really powerful question. Could restoring that tension be the path to a cure?